Okay, so today this is going to be our presentation on the microbial reconstitution versus maternal diet-induced social and synaptic deficits in offspring. It's a deconstruction critical analysis presentation. Hi, so the subject matter of this article is the research into the brain-gut microbiome axis and how it plays a role in the social behavior of mice. And this is done through the use of offspring from female mice fed a high-fat diet, which was found to induce synaptic changes and reduce and the reduction of social behavior. In this article, they made 4 core findings. The first was that the maternal high-fat diet results in offspring results in a behavioral change. The second is that there was a shift in microbial ecology, resulting in the dysbiosis in the gut. And the third was that the maternal high-fat diet offspring have reduced synaptic plasticity in the VTA, which is a part of the brain which will be explained later, and reduced oxytocin production. And the fourth was using metagenomics. They identified a single strain of bacteria that corrects oxytocin levels and the social behaviors in the offspring. And doing all this, their findings, they causally linked maternal diet, gut imbalance, VTA plasticity, and behavior, and suggest probiotic treatment that may relieve behavioral abnormalities in neurodevelopmental disorders. Yes. Yeah, so why is this relevant? Well, it's relevant in our society as maternal obesity is on the rise, obesity in women, and an understanding of the mechanism involved with these disorders would be essential at this time. So part 1, maternal high-fat diets induce social alterations in offspring. So first of all, to establish these diets, they gave for the regular diet, they gave them a diet primarily consisting of carbohydrates, then protein, then a little bit of fat. For the high-fat diet, it was primarily fat, and then smaller amounts of protein than carbohydrates. They made another experiment, reciprocal social interaction task, which was a measure of sociability. They put the mice in a place they'd not previously been habituated, and then put with a stranger, and then their interaction would be recorded, and things like how close they were, how long they spent close to each other would be recorded. It would be like jotted down. And yeah, the 3-chamber sociability task was a measure of sociability. And basically, what would happen? They allow the mouse to habituate itself for 10 minutes in the original zone, and for the next 10 minutes, they'd put one novel mouse in the inanimate object, and for the next stage, they'd put the original mouse and then a novel one, and they'd mark how long they'd be interacting with the different sections. Then the marble burying assay was a measure of repetitive behavior, and it was seen as, and it's known that when mice bury, there's a link to anxiety-like or OCD behavior. And in this experiment, they counted any ball that was 2/3 buried as buried. And in this last test, the open field assay, it's a measure of changes in anxiety-like behavior, where mice could roam free and their position would be monitored. And the more that they'd spend in the outside edges of this box, where the light would be shining here mainly, would show like a level of anxiety. So what was the data obtained from these experiments? Well, from the social reciprocal interaction, it was found that the high-fat diet offspring had fewer reciprocal interactions. That can be seen clearly here by the data shown. In all cases, they had a significantly lower, for example, interaction frequency, like number of contacts, than the regular diet, offspring of regular diet. And then similarly, in the 3-chamber test, these results were also replicated. And this showed that the high-fat diet offspring had reduced sociability and showed no preference to social novelty. And this can be seen here by these actual recordings of where the traces of where the mice went. And you can see that there's not much difference between the inanimate object and the novel mice. And the sociability of the mice is defined as the subject spending more time in the chamber containing the novel mouse than the inanimate object. But yeah, as I showed here. And you can see that in the regular diet that it's a lot less. And yeah, this is the more data. And so the marble bearing assay, what results did they get? They found that it couldn't even be changed by a process called co-housing, which is capable of increasing social behavior by allowing the transition of bacteria from the regular diet to the high-fat diet offspring. But the number of marbles that are buried is associated with anxiety or obsessive compulsive disorder, as I said earlier. And the fact that it cannot be changed by co-housing suggests that the behavior doesn't depend on the changes in the gut microbiome, this burying anxiety, like this repetitive disorder. And the GF in this diagram actually describes, it means germ-free, which was a separate part of the study talking about the gut microbiome being free of germs and how they measure that too. So yeah, the last experiment, how data obtained from the open field assay. It can be clearly seen here that the maternal high-fat diet they spent a lot less time exploring the center, and this is deemed as more anxious behavior as they tended towards the edges. And in this diagram, you see the orange bar represents when the bacteria L. reuteri was added, and it had no significant effect on this behavior, and it shows that this also isn't dependent on the gut. So what did all these tests show? They clearly demonstrate that this maternal high-fat diet does have an effect on their sociability and their behavior. So what do I think that they could have done differently, or like what I thought about it? Well, the main thing I did notice from the start is the fact they used plunger plots to represent their data, which just isn't ever a good idea because of the fact that plunger plots conceal most of your data, the spread, the distribution, outliers, and sample size, which is something that you shouldn't really have when you're presenting such a high amount of data in an experiment. And then another thing with their method I found was that there's a lack of information about the illumination during interaction tests, as the high levels of light can affect the overall level of interaction, the same as the unfamiliarity with the test environment. These raise another question to me, that if these effects of reducing the overall level of interaction improportionately affected the high fat diet offspring and the regular diet offspring, and if they had accounted for those things. An interesting thing I found is that they have performed 2 assays that are included in behavior phenotyping when people are trying to link the genes to the phenotype, and if they choose one more testing the communication of the mice, they could have had comparable testing to those that are investigating that. But overall, it was a very understandable explanation, and the experiments are relatively well explained and shown. And now I'm going to set you over to part 2.
A variety of factors contribute to the cause of abnormal social behaviors within mice offspring with maternal high-fat diets. Maternal obesity has shown to alter the gut microbiome of offspring. In order to examine whether maternal high-fat diets cause alterations in offspring gut microbiota, an analysis of bacterial composition and community structure in the feces of regular and high-fat offspring was performed using 16S ribosomal RNA gene sequencing. 16S rRNA gene-targeted PCR is commonly used for detection and identification of bacterial pathogens. It amplifies all the 16S ribosomal RNA from all bacteria in the community. It can then be used to create a trace to portray the abundance of each bacteria in the community. It was found that there was not a difference in microbial diversity within the gut of regular and high-fat offspring when computed on a weighted unifrac. However, there was a difference when computed on an unweighted analysis of unifrac. Weighted unifrac takes into account the relative abundance of species and taxa shared between samples, whereas unweighted unifrac only considers presence or absence. Weighted is more useful for examining differences in community structure. Unweighted is more sensitive to differences in low abundance features. Within the article, both were shown. This prevents bias as the authors did not just show the results which support their hypothesis. As shown in the graph to the left, the gut microbiota of mice with maternal high-fat diets seem to have less diversity. However, below in the p-values, the paper states there is no significant difference. This data is presented well, particularly B and D, which show a visual comparison. Mice are coprophagic and are able to transfer gut microbiota by the consumption of each other's fecal matter. The study examined if housing regular maternal mice with those of high-fat maternal diets would prevent social deficits within maternal high-fat mice. This was investigated by housing a maternal high-fat offspring with 3 regular maternal mice. Control groups consisted of individual cages containing either 4 high-fat or 4 regular mice. Fecal samples were collected, and social behaviours of both offspring was assessed when mice were 7 or 8 weeks old. Maternal high-fat mice co-housed with maternal regular mice showed typical social interactions. They also showed typical sociability and preference for social novelty as determined by the 3-chamber test. Therefore, co-housing the control mice adjusts the social deficits in maternal high-fat offspring. Next, the microbiota of those co-housed was examined. High-fat mice housed with regular offspring did show a shift in bacterial phylogenetic profile which resembles a healthy gut biome, similar to those of the regular maternal mice. The bottom graph shows that co-housing 1 regular mouse with 3 high-fat offspring was sufficient to adjust the microbiota phylogenetic profile of the high-fat mice. Germ-free mice have shown that intestinal microbiota can influence brain development and function. The paper hypothesized the lack of the same bacteria in germ-free as in maternal high-fat diet is the cause of atypical behavior between both sets of mice. To investigate this, fecal microbiota from regular and high-fat mice was transplanted into 4-week and 8-week-old germ-free mice. Germ-free mice who received fecal microbiota from regular mice showed typical social behavior. Those 8 weeks did not. Germ-free mice which received fecal microbiota from high-fat mice did not show any typical social behavior. Bacterial communities in germ-free mice that received fecal microbiota from high-fat donors clustered differently from those who received the transplant from regular mice, regardless of if the transplant was at 4 or 8 weeks. This suggests there is a window of time of up to 4 weeks for neurodevelopment when introducing microbiota in order to improve social behavior. To investigate which bacterial species are absent in the microbiota community of maternal high-fat diets, metagenomic shotgun sequencing was performed using fecal samples from maternal high-fat offspring and regular offspring. All extracted DNA is sequenced and then the genes within the sample are determined and assigned to bacteria. The abundance of several species of gut bacteria was lower within maternal high-fat offspring, more specifically L. reuteri. It is thought that decrease in L-reuteri within maternal high-fat diet was responsible for the atypical social behaviour. This was confirmed by the study in which L-reuteri is given to mice through drinking water. It would be interesting if they used L-reuteri the same as they did with the fecal transplant in the previous study and compared the results to see if the 4-week neurodevelopment window was still there and if the transplant did not show a change in the anxious or repetitive behaviours observed as the L-reuteri experiment did.
Thank you, Chloe. I'm going to be focusing on atypical synaptic plasticity and the difference in oxytocin production of MRD and MHRD offspring. The ventral tegmental area, the VTA, is involved in cognitive and emotional processes and has a major role in reward and pleasure pathways. It is found in the midbrain and is characterized by an abundance of dopaminergic neurons. As one of two major dopaminergic areas in the brain, the other being substantia nigra, there are two pathways which extend from VTA to other areas, the mesocortical and the mesolimbic pathway. The mesocortical pathway corresponds to the cerebral cortex, and this is responsible for motivation and emotion. The mesolimbic pathway transports dopamine to the nucleus accumbens and amygdala and is associated with reward. So dopamine signaling along this pathway is important for processing rewarding experiences. Oxytocin is a neuropeptide involved in social behavior. Its release acts as a positive reinforcement for social behaviors. To test the function of oxytocin in social situations, participants taking oxytocin through a nasal spray showed an improved ability to understand emotional information through interpretation of facial expressions compared to participants who did not take oxytocin. In rats, administration of oxytocin increased their mother-like behaviors to baby rats, which were not their own. Therefore, this neuropeptide is a good indicator of social behavior, as increased levels of oxytocin seem to be correlated with high levels of social interaction. Decreased oxytocin levels in MHFD offspring. As oxytocin is mainly synthesized in the paraventricular nuclei, the PVN, which can be found in the hypothalamus, Buffington et al. Compared the oxytocin expression levels between MRD and MHFD offspring. As a result, he found significantly lower levels of oxytocin immunoreactive neurons in MHFD offspring, as well as a lower oxytocin immunofluorescence intensity. The reason for decreased oxytocin is not due to MHFD offspring having a lower number of PVN neurons, as Noon staining showed that there was no significant difference in the number of neurons between the two types of offspring, as shown on the right here. Noon staining is a neuronal nuclei staining where antibodies are used to identify mature neurons. So this shows that there are lower levels of oxytocin in MHFD offspring, which could be associated with their social behavior. And Buffington tested this further by the direct administration of oxytocin to the mice, and this was found to encourage social behavior of MHFD offspring, as they spent more time with the stranger mouse after taking oxytocin. So atypical synaptic plasticity and long-term potentiation. The mesolimbic pathway is activated through experiencing rewards, and this corresponds to the VTA, where information is transmitted from oxytocin-expressing neurons in the PVN. Oxytocin activates VTA neurons in mice and humans to help process social cues to either promote or discourage social behaviors. And long-term potentiation is a type of long-term plasticity which enhances synaptic strength. And this means that these synapses elicit a stronger electrical response to stimuli. This long-term potentiation is indirectly measured through activity of VTA dopaminergic neurons by AMPAR and NMDAR ratios, which are glutamate receptors. So to study the difference in long-term potentiation between MHD and MFHD offspring, Buffington investigated the effect of social interactions on long-term potentiation, and he compared the ratio of glutamatergic excitatory postsynaptic currents in MRD and MHFD offspring. And this was after 10 minutes of social interaction of the MRD offspring and MHFD offspring with a stranger or familiar mouse. As a result, the LTP in the VTA dopaminergic neurons were only observed in MRD offspring interacting with the stranger mouse. MHFD offspring showed a lack of LTP in VTA after interacting with both the familiar and stranger mouse. And this is shown by the blue figure on the right. So differences in AMPAR and NMDAR ratios were only significant between stranger interactions with the familiar mouse and the baseline ratio in MRD offspring, as shown by the significant p-value here, as it's less than 0.05. However, the non-significant p-values for MHFD offspring indicate that AMPAR and NMDAR ratio across all conditions were not significantly different, which means there was no long-term potentiation in their VTA. And there was also no difference in their basal synaptic transmission across MRD and MHRD offspring. So the observed LTP from the higher ratio is due to variation in the mesolimbic dopamine reward system. Behavioral patterns also match the electrophysiological results, as MRD offspring spent more time with the stranger mouse than familiar mouse compared to MHFD offspring, which demonstrates their preference for social engagement. Therefore, social interaction only produces long-term activation of dopaminergic reward pathways in MRD offspring and not MHFD offspring, which suggests that they have an atypical synaptic plasticity. To investigate this further, I looked for a study to find a factor which affected synaptic plasticity in mice, and this led me to a paper by Lee et al., who found a candidate which may be involved in this process. CHST14 regulates neurogenesis of neural stem cells. They examined various proteins with functions associated with synaptic plasticity and found that a lack of this sulfotransferase significantly reduces the expression of these proteins related to synaptic transmission in the hippocampus. So LTP in the hippocampus was reduced in mice without CHST14 compared to the wild-type mice. It would be interesting to see if mice with ASD have this sulfotransferase to see whether there is a correlation between the CHST14 and atypical social behavior. Thank you.
Okay, so I'm going to be talking about how Lactobacillus reuteri treatment restores oxytocin levels, VTA plasticity, and social behaviours in mice. So, as previously outlined, MHFD offspring are deficient in several bacteria, including L. reuteri, which impact on their social behaviour. These mice had their drinking water treated with L. reuteri. This restored both social behaviour, with the treated mice having a preference for social interactions over empty cages, as figure 4b shows here, and the preference for novel social interactions over familiar mice, which is shown here in figure 4c. Interestingly, heat-killed L. reuteri did not restore behaviour, it must be live, which is shown here in these darker orange columns. These changes are specific to one species of bacteria, as treatment with the similar species, L. johnsonii, which was also reduced in MHFD offspring, had no effect. This raises the question, if murine social behaviour is restored by only one specific species of bacteria, how transferable is this to humans? Also, in the vehicle control, only 3 mice were used, as is shown here in the key. This does seem a very small sample size to use. The paper does not discuss the extent to which social behaviour is restored. Figure 4c shows that it may even cause slightly greater social interaction than untreated maternal regular diet mice, which is on the far left here. Is this desirable? There is no hypothesis anywhere in the paper as to the mechanism of action. If bacteria must be live, is there a specific protein or metabolic process that has these effects? I feel that this might need developing. Although there is no reference to the mechanism used by L. reuteri, the authors believe oxytocin is involved in the process. The number of oxytocin expressing cells in the paraventricular nucleus, or PVN, was recorded, and MHFD L-reuteri treated offspring had a greater number of oxytocin expressing cells than in control MHFD offspring, as you can see here in figure 4.0. As expected, there was not only a greater number of oxytocin expressing cells, but also a greater quantity of oxytocin present itself. Figure 4p shows that oxytocin fluorescence intensity rose from around 31 to 40 arbitrary units in mice treated with live L-reuteri over heat-killed bacteria. Interestingly, MHFD offspring only showed a deficit in oxytocin expressing cells and not in total neuron count, which shows the oxytocin pathway is the central metabolic process here. Just below, I've created a diagram summarising the findings of the paper to this point, as I feel that a summary was lacking from the paper. L-reuteri reconstitution causes a rise in oxytocin expressing cells by an unknown mechanism, in turn restoring social behaviour. From these outlined experiments, the authors concluded that oxytocin immune reactive neurons in the PVN are restored by L-reuteri treatment. I would like to note here that the oxytocin experiments had a sample size of only 3 mice per category, as you can see here. Although there will be ethical constraints, this is a very small sample from which to draw clear conclusions. Oxytocin neurons, which as we've seen are restored by L-reuteri treatment, project onto the VTA in the brain and activate it, which can be rewarding. No VTA stimulation means no reward from social behaviour. To analyse if social interaction gives long-term potentiation, or LTP, of VTA neurons, AMPA receptor to NMDA receptor ratios were recorded 24 hours after a social interaction. Here, the paper gives no description of what we might expect, but following research, a higher AMPA to NMDA ratio is associated with LTP and reward pathways. An increase in this ratio from the baseline was seen in L-reuteri treated mice, as you can see here in figure 5J, suggesting reward pathways were activated as a result of oxytocin stimulation due to L-reuteri supplementation. As Tong has covered, the MHFD mice not treated saw little to no increase in this ratio following interaction with a stranger. This suggests the L-reuteri treatment has had a substantial impact. In parallel with this, we can also see in figure 5H the spike from the baseline in LTP caused by interaction with a stranger for L-reuteri treated mice. Just wanted to note here that control bacterial species were used to examine the specificity of the social effects of L-reuteri, but not of oxytocin expressing cells or AMPA receptor to NMDA receptor ratio. It would be interesting to know if these elements are affected by the other bacterial species affected by HFMD. There is no diagram in the text showing the findings of the paper as a whole. Figures were entirely graphical and scattered through the text, often with legends separated by a page of writing. Some figures integral to the understanding of the text and its findings were also found in the supplementary section. Bringing that all together, here is a diagram of what the findings appear to be. Social interaction or oxytocin alone failed to restore VTA-LTP, but both together did. L-reuteri did accomplish this, which suggests oxytocin expression and social interaction have a synergistic effect on LTP and therefore on reward mechanisms. L-reuteri generates both oxytocin expression and the desire for social contact, and so restores LTP. It could be that L-reuteri creates oxytocin expressing neurons by an unknown mechanism, which in turn creates a desire for social interaction, which in turn stimulates LTP and reward pathways to reward social behaviour. The paper has been criticised at several points throughout the presentation, but here are some of the downfalls I haven't yet mentioned. Some description of how the microbiome interacts with the brain would have been helpful, as it is the focus of the paper. Cryan et al. Have a great explanation of this, which I've used to produce this figure, outlining the varied roles of the gut-brain axis. It's been found that infant microbiomes are a direct result of fecal and vaginal maternal transfer, which the paper doesn't discuss. If infants are L-reuteri deficient, does this mean that the mother is? And if so, are the obese mothers behaviourally affected? This isn't addressed by the authors, and if this is the case, It may be that microbial reconstitution of mothers with high-fat diets could antenatally provide offspring with the necessary microbiota. The paper asserts that fecal transplants don't reverse repetitive behaviours associated with ASD, only the social ones, so they don't depend on the microbiota. This has not been supported anywhere in the paper, as far as I can see. And the paper also seems to assert that co-housing restores social behaviour by fecal transmission of bacteria, for example, L. reuteri. There is no mention of the fact that environmental or social mixing with non-HFMD offspring may be the largest factor here, so this could have been explored. So, in summary of this section, more research and at a greater scale is needed to secure the findings of the paper. The specific metabolic mechanisms L. reuteri acts on must be determined, and we're also lacking any information on the degree to which the findings concerning L. reuteri and the murine system may be relevant to humans. Okay, thank you. I'll pass on to Orzor, who I think is doing the final part.
Hello, and this is part 5 of the paper. We're going to look at the summary and the critique of the paper. So we start with the purpose of the author. So the author wrote this paper to determine if diet is capable of altering the behavior of the offspring, mainly whether the lack of key bacteria affects the offspring's gut microbiome, and whether that since they lack the microbiome, the proper microbiome, whether this causes the offspring to be socially impaired. But the author also wants to find out whether these changes can be reversed somehow. So as the abstract shows, whether we can introduce bacteria that will improve the social behavior of the impaired offspring. So we start with the highlights, where we find the key findings of this paper, which is the author starting the experiment by using mice instead of humans to prove the association between the high weight gain in female mice and whether the offspring inherit, whether the offspring tend to lose certain key bacteria due to this weight gain and become less socially impaired in a way. So the findings in the paper support the author's theory and reveals a link between the maternal fat diets and development of neurological disorders such as autism within their offspring. So the author compares the difference between the female mice with normal diets and the high fat diets, which the paper goes in a lot. So, but it also reveals that through the reintroduction of key bacteria, the mice can regain their social skills. So the introduction starts with showing the background of the paper, mainly with focusing on the research behind neurological disorders such as autism and high-fat diets, but they also focus mainly on the human side, and they explain how the rise in obesity among mothers may be linked to the rise of autism among offspring. The author also notes that those individuals with autism tend to have gastrological and diabetes access issues as well. So the results are various. There's various results tend to support the author's argument, with high-fat diet offspring tending to have fewer social interactions and perform worse socially compared to their counterparts. They also explain how the gut microbiome of the high-fat diet offspring is reduced, and they contain, and certain species are missing within the microbiome, which may show that these bacteria are essential to regain social behavior, but co-housing may be a possible treatment due to how it leads to these offspring regaining their social behavior while living with normal mice. So the author notes this and discusses how this might work, that how certain bacteria are capable of improving social function in mice, and because of they improve social function, these mice behave better socially, and high-fat diets and overall obesity affects how these bacteria develop in offspring, certain offspring, which leads to offspring losing their social skills as they develop, and also developing neurological disorders. So while we have a lot of research behind that, it is not enough to determine the biological mechanism of how these disorders develop. So as we talked about a lot about the paper, but what did the author do wrong? And we could say that their language wasn't great, especially how they critiqued obesity levels in women and describes it as an unfortunate condition, as well as describing those with autism as defective, especially when they refer to the mice. But also the research feels incomplete since it misses a method section which details how they performed the experiment, but also they also didn't explain much about how the high-fat diets affect on people's minds, or how even though the research is focused on humans, they use mice. So the author uses pure speculation to basically determine whether the results can be replicated with humans, even though they haven't done the experiment yet. So thank you.